This is part two of the chapter 11 lecture notes, and we had left off on metaphase one of meiosis one. Um, and the only other thing I wanted to mention, the main thing that happens in metaphase one is that instead of the chromosomes lining up single file at the metaphase plate, the homologous chromosomes, the homologous chromosome pairs line up at the metaphase plate. So in humans, there would be 23 pairs of chromosomes lined up at the metaphase plate. And then if you look down at the second point, maternal and paternal chromatids orient randomly and are mixed when they migrate to the poles. That random orientation, in other words, sometimes the chromosome that came from the father will line up on the left, and sometimes it will line up on the right. So that will determine what combination of chromosomes ends up in the gametes. So this random assortment of chromosomes, this random lining up of the chromosome pairs, is going to increase genetic variation. So far, we have talked about two things that create genetic variation. So one is crossing over during prophase one, and the other is the random lining up of the chromosomes in metaphase one. It's actually got a name, and we'll talk about it. I'm going to write it at the bottom. It's called independent assortment. Sorry. It's called independent assortment. That's an N. Okay, independent assortment, and that is just that the chromosomes line up randomly in metaphase one. So sometimes in the chromosome pairs, the one that came from the father, maybe will line up on the left, and sometimes maybe it'll line up on the right, and then you go to the next pair, and the same thing can happen. So you just have this mix of um, chromosomes that randomly lined up, together at the metaphase plate. In anaphase one, instead of the sister chromatids pulling apart, the tetrads, the microtubules pull the tetrads apart, or you could say the homologous pairs apart. And then in telophase one and cytokinesis, the separated chromosome pairs arrive at opposite poles. And cytokinesis, if the cytokinesis actually occurs all the way, the nuclei do not reform. So this is one difference. Because there's going to be a second division, the nuclei don't reform around the groups of chromosomes. Now, this shows you a difference in the plants versus animals in cytokinesis, but it's very similar as what we talked about in mitosis. There is a cleavage furrow in animal cells and then a cell plate that forms in plant cells. This animation is on slide 14. I would encourage you to watch it. Slide 14. And now we're going to move on to the second division called meiosis two. So we're going to start out with the two daughter cells that were produced in meiosis one, and each of these daughter cells have now a single set of chromosomes that still contain sister chromatids attached at the centromere. So what's going to occur in the rest of these stages of meiosis two is going to be identical to mitosis. In prophase two, you will see the chromosomes appear. They may have gone back in the form of chromatin, or they may not have, but the chromosomes will be visible. There will be no nuclear envelope. The centrosomes will form, and the spindle fibers will emerge from the centrosomes, and the centrosomes will move to opposite ends of each of these cells. And but that's it. So then we have prometaphase two, where the spindle fibers will attach to the kinetochores identical to mitosis, and start moving the chromosomes to the metaphase plate. In metaphase 2, they're going to be lined up at the metaphase plate, this time in single file. And then in anaphase 2, the sister chromatids will separate, just like in anaphase of mitosis. And telophase 2 and cytokinesis will be identical to mitosis. Nuclear envelopes will reform around the chromosomes, and the only difference is what you end up with. So at the end of meiosis 2, you will have 4 haploid daughter cells, whereas at the end of mitosis, you had 2 diploid daughter cells. The notes on the stages of meiosis 2, it says in prophase 2, the events do depend on species-specific differences. Sometimes the chromosomes decondense at the end of meiosis 1, so they form back into chromatin. So the chromatin would need to condense and form back into chromosomes. But some species, this doesn't happen, so the chromosomes never decondense. In prometaphase 2, the sister chromatids have kinetochores that are attached to the spindle fibers or spindle microtubules. Same thing that happened in prometaphase of mitosis. And this is just a drawing of the lining up of and the separation of chromosomes in meiosis 1 versus in meiosis 2. So this is good for you to see. In prometaphase 1 of the first meiosis, remember that the homologous pairs have found each other and matched up and formed tetrads, and that process was called synapsis, and that happened in prophase 1. So in prometaphase 1, those pairs are moving toward the metaphase plate, and you can see what they look like here. And then in metaphase 1, crossing over occurred. So in anaphase 1, this is what it looks like when the homologous pairs pull apart. You have crossing over that occurred between the sister chromatids that were close to each other, one sister chromatid from each of the homologous pairs. They are actually called non-sister chromatids because these two were not sister chromatids. They came from separate chromosomes, so they're called non-sister chromatids. But the point is, we can see where crossing over happened in metaphase 1, and now anaphase 1, those homologous pairs are being pulled apart. Then we get to meiosis 2, where the individual chromosomes with the sister chromatids line up single file at the metaphase plate. So in prometaphase 2, they're lining up like this, and it is the kinetochores that are being attached to the spindled microtubules or the spindle fibers. And then in anaphase 2, it is the sister chromatids that are pulled apart. In metaphase 2 and anaphase 2, the events proceed as in mitosis. Sister chromatids separate, move toward opposite poles, and telophase 2 and cytokinesis are identical. The only difference is the end. At the end of cytokinesis, there are 4 haploid cells, and they are not